All right, so Lance was kind enough to let me pirate his account for a moment so I could show you guys this, because we've been running into some difficulties, and I helped one of your other classmates today. Most of you guys at this point have had to pay for it, right, or getting pretty darn close to having to pay for Wiley Plus. Lance, you probably already paid for yours, huh? But one of the other students, I'll tell you where it showed up for her. So I've went ahead and added, just to make it a little more streamlined for you guys, this link right here that says Wiley Assignments. I hadn't added that before because the videos that I watched for training told me not to. But another instructor yesterday I was helping, and she did turn it on. I thought it would give you guys access to our side of the assignments, right? But apparently it knows whether it's a student or an instructor that's clicking on this link. In the instructor resources, I'm clearly not going to give you a link to, although I probably could. Oh, yeah. What did I say about the deadlines, y'all? They're guidelines. It doesn't shut down. You can still do it after it. And I don't have it set where it'll take points off if you do it after. But, you should get it done by that date. Does that make sense? Okay. Is she good? Yep. Right. We're going to cover that next. Okay, so I added this link, but what I've been doing is I will put the links in the modules. But I'm guessing by the titles, too, it's not really cluing you guys in that that's your assignments. I think, is that the disconnect there? Okay. So I added this link, so when you do click on it, it will actually bring you to the assignment page in Wiley Plus, where all the assignments will be listed. And then you can just click on the assignment. So he hasn't done this one yet, right? And so you could click on it, and you can see, right? Then you click next. Now right here, over here, for the student whose one was getting ready to expire, it said she had 1 day left of the grace period, and then right next to it, it had a button or a word that said register. And when you clicked on that, then it took you to the other screen where you click to pay and register. And everything went smoothly with that? You finished it up? Okay, cool. All right.
Is that where we answer the homework?
Yeah, this is a homework assignment. This just tells you the particulars about it, right? That you have unlimited attempts. After the 5th attempt, it'll show you links. It'll never show you the answers. Study objectives have been enabled. It is graded, and when the due date is. But yes, attempts after the due date will be marked late, but not taking points off, right? These aren't bonus. These are required assignments. Yeah. This is the actual questions, right? So you can click on them, and it'll go right to them, or you can just keep clicking next to move throughout the assignment. You haven't done the first assignment yet? Well, clearly not, because I should do tonight. Okay. All right. I hadn't spied on you guys yet, so I didn't know. But some people have done it, so some people have figured it out. Yay. Right. I keep the highest grade. Yeah.
But it doesn't reset the questions, because I went back and I was like, let's try and take it again. And questions are still...
What do you mean?
Like, if you got them all right, they all have green, and it doesn't reset the question like...
That you can take them again?
Take it again.
Shit. Okay. No, thank you. Because I don't know. Like I said, this is a new system for me. I'm going to call the book rep today, tell her to get her ass here next week and train us. Because the training she had, they weren't ready for us. They didn't have our stuff set up. And we've been trying to learn through the videos, and we're running into problems. And, it has a lot of great stuff for you guys, but, you and I and all of us have got to learn how to use it.
It used to be where you would say you missed a question.
Yeah. Yeah, and that's the thing is I don't understand all these settings to set it how I really want it to be for you guys. So I'm going to have to play around with it. Huh? Until you got it right, yeah. What good is that going to do you? Okay. All right. So let me log out of Lance's and pause my record. So watch that animation, right? That can be helpful. See if you can fill out this table on your own, and then, of course, look at the answers. And we're going to talk about gram stain like forever, like all semester long. So this is one of these concepts that I always tell my students, like if you come to me at the end of the class and you still don't understand it, you might actually see me cry. Because it's that important. Right? So I won't ever shut up about it. So at some point, hopefully, it will sink in. Okay, so we're going to go to today's lecture, though, which is Prokaryotes, which is covered in both Chapter 2 and parts of Chapter 4 I included in this lecture. And so last time I had showed you guys how there's a podcast for the opening picture in your book for this, and it even has a transcript so you can read it. You don't have to listen. So our first objective as it relates to prokaryotes is be able to identify the various shapes and sizes of prokaryotes. So we use some terms, right? What shape does Coccus refer to as? What's bacillus? What's spiralium? Rectangular, I hope I don't have to explain that to you guys, right? Okay. And square? We're good? 4 equal sides? Yeah, square. Okay. And then some of them, after they divide, they actually stay next to each other and form characteristic arrangements. So cocci are the ones that do this the most. So you'll notice mostly we have the cocci arrangements. And I have a good summary slide in this presentation that goes over all of these. So we're going to go through these step by step. But you know, this is of course what you need to be able to know and recognize for the exam. So remember I said even if you don't want to read your book, at least listen and watch your book, right? These 3D animations are awesome. And if you click on it in presentation mode, it will go to the internet, so you got to be connected to the internet, and it'll go right to that animation that goes over what do bacteria look like, right? So spherical, and so remember these are 3-dimensional organisms, right? Even though we look at pictures and we look at them under the microscope and we really can't see the depth of them, you can in these pictures a little bit because these are scanning, remember we learned last time, SEM, scanning electron micrographs. And of course, again, all of them have been colored, right? They've gone and made them pretty colors. So ones that are spherical, like a ball, are called a coccus if you're looking at a single cell. If you're looking at multiple cells, like in this picture, these would be cocci. This is Latin. And for me, how I remember plural from singular, it kind of is the opposite of what we're used to. When we say I, we're talking about a single thing ourselves, right? In that case, it's actually plural, right? When it ends in I, it's plural. When we say us, US, right? Us, we're talking about a group. Where in this case, US, that ending to a word, is singular. It refers to a single cell. So rod-shaped bacteria are sometimes referred to as bacillus, or if you're talking about all of them that you see in this picture, bacilli. But quite honestly, a lot of instructors and scientists don't actually use this term. Part of the reason for that is it's also a genus name. You guys already learned about an organism whose first name is bacillus. Second name is anthraxis, right? Causes anthrax. So it can get kind of confusing. And as you can guess by its name, guess what shape it is? It's rod-shaped. So a lot of scientists actually just say rod-shaped when they're referring to shape, so people know we're talking about shape, as opposed to bacillus, which is also a genus name. Now, if we were writing out that name, how would you know that we were talking about the genus as opposed to the shape bacillus? It'll be what? It'll be a capital B, but also if it was the beginning of a sentence, it'd be capitalized, right? We underline if we're handwriting proper names of organisms. If you're typing it, you italicize it. So it would be italicized, right, if you were reading it in a text. And that would kind of give you a clue right away that they're talking about the name, the genus, that group of organisms, as opposed to just its shape. But again, these two kind of go hand in hand. All the bacilli genus in that genus are rod-shaped. But there are other organisms out there, like Escherichia coli, E. coli for short, right? It's rod-shaped, but its genus name is Escherichia, not bacillus. Make sense? So again, that's why typically we'll just say rod-shaped. Makes it easy. No confusion. And then some of them, instead of being a straight rod, they have a curve to them. They almost like look like little comets. So you see the curve there? Those are referred to as vibrio or vibrios, the normal plural we're used to add an S. Thank you. But you can definitely see here too, you see how they're next to each other in this first picture. You see how they're forming chains? These are actually cocci. They're really tiny, but they're forming chains. These guys are forming chains. They're next to each other, right? There are 3 of them. This is probably getting ready to become 2. It's really long. You can even see some pairs here, right? Where they seem like they're staying together, maybe even forming chains in this picture. So this lovely diagram I borrowed from the internet does an awesome job of showing all these arrangements that cocci can form. Cocci are the ones that have the most arrangements, where they stay next to each other in these characteristic groupings after they divide. So some of them, when they divide, they'll stay together in pairs. We refer to that as diplococcus. Coccus for the individual shape of the cell. Diplo refers to 2. Anyone know a pathogen that is cocci and it's in pairs? It's a diplococcus. They look for it in your spinal fluid when they take spinal fluid and they're looking for a particular type of meningitis. No. Meningitis. Neisseria meningitis. Gonorrhea is also a diplococci. So under the microscope, when they see 2 cocci next to each other, and even more if they're gram-negative, they're like, oh, it might be this organism. They'll do, additional testing. And then there's one that we really don't like. Right? This is the next one, Streptococcus, because it causes an infection. One of the species in this genus, because this is another one where it's confusing, right? Streptococcus is an arrangement, but it's also a genus name, right? And we abbreviate this genus name for an infection you can get in your throat called strep. That's where it gets that name. It's Streptococcus prurigens that causes this infection. And under the microscope, you'd see cocci forming chains. So I always kind of remember for this one, most of us have had strep throat, right? If you haven't, all the rest of us are jealous. You don't even want to swallow, right? It's so painful. Your throat is just like on fire. So I always imagine this chain of bacteria and about how I would not want to swallow a chain. And your throat is almost like a tube, like a chain, right? So I just, I envision strepto, I envision a chain, I envision my throat, and how I certainly would never want to swallow a chain when having strep. So strep, that prefix means chain. Tetrads are another thing that can happen. And what does tetrad mean? It means 4, right? So it's a square group of 4. And one of the organisms we'll look at in lab this week, Micrococcus luteus, forms these characteristic tetrads. It's really cool. Look at it under the microscope. Sarcinia, which unfortunately again is another genus name, and guess what the organisms look like? They form these packets, right, of 4 cells on 4 cells, so it's like a cube. Which of course is not easy to see with a light microscope, huh, because they're stacked on top of each other. They really didn't get a good idea for this type of arrangement until the more powerful phase contrast microscopes and electron microscopes, and being able to do the scanning electron micrography. They could see that they were forming these packets. And then there's the last one that most people confuse with strep all the time, right? Staphylo and strep, students and people switch up the names all the time. Staphylo refers to a clustering. So these guys divide on random planes in 3-dimensional space, and they form these grape-like clusters. So Staphylo means clusters. And again, we can get an infection of our skin that we abbreviate the name of the organism that causes it, and we say staph, right? It's Staphylococcus aureus that's usually causing that infection. And again, when they say you have a staph infection, they're just abbreviating the name of the organism that causes it. So yeah, some terms, some not so easy terms that you're going to experience a lot in microbiology. So do the best you can. If my little stories help you remember between strepto and staphylo, yay. If not, make up your own story. If you want, you could share it with us too.
So you have to write them on a test to abbreviate them?
You don't write on my test. Remember, it's multiple choice, matching, true and false. Just like your homework. The 12th of September, clearly. Okay, so your book doesn't really talk about these guys in the shape category. They do mention, I thought they did. No, not on this slide. So I put it before it. So these are spirochetes, and we'll talk about these guys a couple of times. These guys are really cool. Under the microscope, they look like these really tight zigzag lines. If you were to look at them in 3-dimensional space, they're helical. Like a spiral staircase, a slinky, like DNA. Right? And this shape that they have, this spiral shape, is due to modified flagella. They're on the outside of the cell within an outer sheath, like in a kind of a periplasm space. So these little dots are these axil filaments, the name given to these modified flagella, all these little green dots. And this is probably a transmission electron microscope picture. And again, they colored what they wanted you to look at to match their drawing. They've actually engineered spirochetes to not have these axil filaments, and they just become these long rods. So that shape is determined by these axil filaments. And then their ability to have this unique movement that they have, where they corkscrew through their environment, is due again to these axil filaments. So we don't work with any of these in lab because they cause things like syphilis and Lyme disease. So yeah, not playing with these guys. Others that have this really cool shape are symbiotes that live in the stomach of cows and other ruminants, and they actually help break down the cellulose that cows eat. Cows don't actually eat grass. The microbes in their gut eat the grass, and then they eat the microbes. So if you give antibiotics to a cow, the farmer gets really nervous, because if you screw up, you could kill the cow. Because if you kill off that cow's bacterial gut microbes, he can't eat, and he'll die. They really rely on their symbiotes. So this is a cool website that Berkeley has, and then under the videos and animations for Module 1, and this one doesn't, this is just, you know, for show, doesn't have any audio. And I'll just play it within here. Have a quick YouTube. That's the one that causes Lyme disease, and see it moving. Isn't that cool? He's wiggling on through, but he's like spiraling, corkscrewing through the medium that they have there. And that's probably phase contrast microscopy, because you can really see them. They're like glowing. Very cool. So, oh, there's spiralium. So others, spiralium or plural spiralia, are not helical. They're like squiggly lines, right? They have multiple bends to their really long rod shaped. And they tend to be quite large bends. Do you see how they are here in this light microscope picture? They're not as tight as you see with the spirochetes, right? Where the spirochetes look like little zigzag lines. Then you have others that don't hold a specific shape, and they're referred to as being pleomorphic. Can we close the door? It's getting noisy out there. Is it locked again? No, okay. Yeah, oh man. That's okay, hopefully everyone's here.
So we can get out. They can't get in.
Yeah, that's all right. We won't let them in. If you're this late, you got problems. Okay, so they don't hold a characteristic shape. So look at this. This is all the same organism in this picture. Wow, there's some funky shapes there, huh? This is a particular organism that causes a type of pneumonia known as walking pneumonia. Because people are up walking around and they don't realize how sick they are until you're like running up 4 flights of stairs with your best friend, doing rounds as a resident assistant in college, and she passes out at the top of the stairs because she overexerted herself and she had walking pneumonia. Right? So again, most of the time you're up walking around, right? When you exert yourself, like running up 4 flights of stairs, you pass out at the top because you can't breathe because you have this infection. So they're not holding a characteristic shape. Why would they? What structure usually helps them hold their shape? The cell wall. So these guys, guess what? No cell wall. So what term do we use to describe these bacteria that don't have a characteristic shape? Pleomorphic. There's a whole genus of bacteria that contain organisms that have these characteristics. Anyone know their name? They're mycoplasmas. And how I remember this one, does anybody know what myco stands for? What mycology is the study of? What microscopic organisms? Fungi. So do you see how these are kind of stretched out? They almost look like fungi, don't they? Almost look like hyphae that fungi form, these long filaments, right? So when they were first looking at it, they probably thought, you know, it's a fungus. And then come to find out, oh, it's a bacteria. So I think they kind of stuck with that myco name. And we used that myco name once else when I was talking about acid fast staining, right? Mycobacteria. Again, they looked like fungi to them. When they grow on agar, they kind of look filamentous. But then when they did more analysis, like, oh, they're bacteria. So in this case, too, how I remember this one, I'm not sure if it's how they got their name, they kind of look like fungi. And remember, they don't have a cell wall, they just have a plasma membrane, just a cell membrane. So mycoplasma, think of plasma membrane, no cell wall. So mycoplasma pneumonia was what causes walking pneumonia. That's what my friend had. The bad news with this, and we're going to talk about later in this topic, is that anybody know what penicillin targets? What does it stop the cell from being able to structurally make? Well, the cells can't make or repair this structure. When they go to divide, they burst. So what helps protect them from bursting? The cell wall. Mycoplasmas, do they have a cell wall? No. So could you give your patient penicillin? Yeah, you could, but it wouldn't do anything for the infection, right? Because they don't have a cell wall, so who cares if they can't make it? They weren't going to make it anyways. Right? So you got to give other drugs that affect other things in the cell. So bacteria, believe it or not, some of them can grow somewhat filamentous. Their cells really stretch out like you see in this first picture. And they form these branches of filaments. So under the microscope, you might think that you're looking at a mold. But in fact, they're bacteria. What would distinguish them as not being eukaryotic, like molds are? What feature do eukaryotes have that prokaryotes don't have? Yeah, no nucleus. And so again, when they grow to large numbers, they look like a mycelia. They look like that mass, right, of fungi that you can see even with your eyes when mold is growing, right? So when you see mold on your bread, right, or your cheese, you're seeing the mycelium or mycelia. If you were to look at it microscopically like you see here, you'd see those long individual threads, the long cells, and multiple cells hooked together in filaments, known as hyphae. So in lab we looked at that, right? We looked at mold under the microscope. And it's beautiful, isn't it? I love their spores. They look like little flowers. Just gorgeous. And then you have others that form these trichomeres. And that's what you see here, where again the cells are stacked on top of each other. They form these long filaments. And they're smooth though. See how they're smooth? And cyanobacteria do this. Photosynthetic bacteria do this. So we can get some really interesting, cool shapes and arrangements to look at under the microscope. For Archaia, which we know are also prokaryotic, but can vary quite a bit from bacteria, they have a whole bunch of them are rod-shaped and spherical and have spirals. Some of them are really irregular in shape, like this guy. Look at this. That's pretty wild. Yeah, I know. Looks like a splash of paint. And then this guy is rectangular. And there are others, like I said, that are square-shaped. And again, these are transmission electron micrographs, right? So these are pictures taken with an electron microscope that they didn't even color, right? They left it in grayscale. So looking at a micrograph, right? It's a photo micrograph, a picture like we've been looking at, or a drawing with structures drawn in it. You need to be able to identify the major prokaryotic structures. So you need to be able to know what they're denoting as inclusion bodies or ribosomes, the nucleoid, plasmids, and the prokaryotic cytoskeleton. Yeah. And the thing is too with this is, y'all, I'm not going to give you just a picture and put an arrow to it, because I think that's kind of evil, because things can be drawn differently, and we're just starting out with this stuff, right? I'll usually give you the picture, and I will give you the description of what it's pointing to, right? So the picture should help you, but you should know the description. Does that make sense? So again, right, if you don't want to read, watch the animation. How many of you guys actually watch these animations? Awesome. They're awesome, aren't they? I wasn't kidding. Okay. So here's an electron micrograph, right? And some of the features are pointed out, right? And then also in the drawing. And I realize this drawing is kind of small. Probably should have blown it up, huh? I can. Oh, yeah. Just Control plus. Where's the plus on here? There it is. No, not in presentation mode, I'm thinking is the problem. But that's okay, because you know what? I know how to do these things. You copy. And you create a new slide. New slide. Erica, hit the right button. And you make it blank. And you cut this baby out. And you put her over there. And then you make her big. Like, bam. Beautiful. Ha
Wow. That's talent.
That's like 10 plus years of using PowerPoint, and I'm still learning stuff. Okay, so we're going to look at all these different features, right? So we're going to, take a tour of a cell as it be, right? So, and for each one of these, too, we're going to talk about its composition. So what do we mean when we say composition? This is not English class. When am I talking about composition in microbiology? What's it made up of? So what are structures made up of? Well, this is a cell. This is a single cell. What are cells made up of? What's the membrane made out of? Lipids and phospholipids, right? But what else? What else is embedded in the membrane? It's the functional component of the membrane. We're going to get to it. Proteins. Proteins run everything, right? If you say protein, you'll be always right. Right? When it comes to cells, it's like 99.9% protein, I think. Something crazy like that. And then, of course, the nucleoid, they already put it there for you. That's DNA. Oh, yeah. Within the membrane and within the cell. Proteins are like everywhere. Like proteins are like the workhorses of the universe. So, composition. Well, DNA is the main part of the nucleoid, right? That's the region within the cell. It's not surrounded by membranes like a eukaryote, right? But they've got to have DNA. But your DNA, even in our cells, isn't all by itself. It's complex with proteins, again, proteins, right, to compact and form what we call chromosomes. And also associated with that is the RNA, right, as it's being formed from the DNA. And that's always happening for the most part. So, and then of course function. Why do we have DNA? Information, right? That's the blueprint for our cells. Yep. So it is genetic information storage and gene expression, because it doesn't just store it, right? It allows us to do things. That information is expressed. That information is encoded, right? It's transcribed into messenger RNA, which is then later translated by ribosomes. Ooh, ribosomes. Every cell's got to have that too, because what do? They make proteins, right? By translation, they are protein synthesis. Guess what they themselves are made out of? Protein and RNA, and actually a special type of RNA. We usually put an additional little letter in front of the RNA. We write a little R to denote its ribosomal RNA. So we're going to go through this list. This is a good summary slide though, right? Of all the information for the most part that you need to know. I feel like okay. I skipped reading one thing. So what's in the cytoplasm, right? It is. Yeah, this whole lecture is called Prokaryotes. This is a general drawing of a bacteria or an archaea. Well, it has a cytoplasm. Yeah. Oh, yeah. So the largest region is actually the nucleoid, right, where the DNA is, and it tends to be centrally located for most prokaryotes, but not always. And that's where the chromosome is, or chromosomes, depending on how many they have, how many long stretches of DNA do they have. And then as I pointed out here, it isn't just DNA, it's RNA and proteins. So the remainder of the cytoplasm is just a stew. You know, it's a liquid matrix, unlike a gumbo pot, right? Very much like a gumbo pot. Everything's in there, right? So you have lots of transfer RNAs, ribosomal RNAs, messenger RNAs, proteins, etc. Within that liquid matrix, there are these structures that we refer to collectively as inclusion bodies. And collectively, they function in storage of large amounts of things. So some of them have particular names to them based on what they store. So this polyhydroxybutanate granules are carbon storage, right, where they pack a whole bunch of carbon in that one region. And they surround it by lipids or proteins, right? They actually literally encase it. Some of them store sulfur that way. Others store gas that way, and it functions not only as storage of gas, but to help create buoyancy for those organisms. So those are aquatic type organisms that have gas vesicles. And this is to allow them to retain gas on the inside that helps them either float up, or if they release the gas, float down in the water column. Or sink down. When you float, you go up, right? Carbiosomes, carboxyosomes, are where carbon fixation happens for usually organisms that are photosynthetic, where they're using light energy to use carbon dioxide gas to form sugars. And these, they have a whole section in your book about these, the magnetostones, osomes. These they believe to be an organelle, right? So it's organized to that level where it's actually encased in membranes, which is a rarity for prokaryotes, right? Because we say one of the key features of a prokaryote is that it doesn't have organelles, especially the nucleus, which is a key defining feature of eukaryotes. So this is one of those ones he almost crosses that definition line for us. So these are some electron micrographs showing you these structures inside the cells. And then they have a whole perspective box in this chapter about these magnetostones. It's really kind of cool, and cool images, and how the organisms are acquiring iron in these little vesicles and lining them up. So they're basically creating their own little compass, and they can find their way based on the polarity of the Earth, right? The magnetic poles and poles of the Earth, and they can help orient themselves in the environment based on that magnetic pole. I mean, they're like microscopic compasses. But they're only able to do that because they're able to line up everything, right? They form this structure and have that iron that's pulled by the magnetic pull of the Earth. So we'll leave internal proteins for next time. But go ahead and take a look at that perspective, right? It's really kind of interesting about those organisms and how they do that.
That's 2.1, right?
Yeah.
Is that the perspective box 2.1?
It's in chapter 2. It's the first perspective box.